Hi everyone, and thank you for joining us today, and welcome to today's Tips and Tricks webinar with Dr. Laura Voita and Vanessa Lambaton on how to choose your super resolution technique. So before the webinar begins, we'd like to go over some quick house rules for the webinar. And I'm sure you've all noticed by now, but you do have a webinar navigation panel on the side of your screen, and this is going to contain a few different sections. So if you'd like to learn more clearly or see one specific section, you can simply just click on the expand icon, which is located in the top right corner of each section. So today's webinar is going to last about 45 minutes, leaving approximately 15 minutes for Q&A at the end. You're welcome to submit any questions that you have throughout the webinar via the chat section, which is located within your panel. However, we will only be answering your questions during the Q&A session. And if we're unable to get to your questions, then we will email you your answers separately via the email address in which you registered with. Within your navigation panel, you'll also find a handouts section. And in here, you can find our company brochure. So please feel free to have a look there or on our website located at www.oni.bio. Please note that today's webinar is being recorded, and within the next few days, you will receive a thank you for attending email with a copy of today's webinar to refer back to. And in addition, a video recording of today's webinar will also be stored on the resources section of our website. And now I would like to introduce you to our presenters for the day, Laura and Vanessa. So Dr. Laura Voita is a PhD in biomedical engineering from Idaho University, and she focused on understanding nanomedicines and how they target cells using super resolution microscopy test techniques such as D-Storm. And in her current role, she is a field application scientist with ONI, and she works to help scientists get started in super res fluorescence microscopy with the nanoimager. And based on her background and extensive experience, Laura can relate to the pain points of getting started in super res microscopy. So she aims to make the technology more accessible with practical training and analysis sessions, including sample preparation, brainstorming, and imaging tips, such as today. Now on to Vanessa. Vanessa Lambaton has an MS in biology from UC San Diego, where she studied the sleep homeostat of Drosophila. In her role as a field application scientist, she draws inspiration from her prior position as an imaging specialist, fueled by a passion for overcoming challenges, particularly in the realm of D-Storm. And she is dedicated to lowering the barrier to entry for researchers. And this commitment aims and aligns with the overarching theme of the webinar, where she intends to share valuable insights on how to choose your super res technique. And with that, I will now pass the presentation over to you, Laura and Vanessa, to begin the webinar.
Thank you, Carolyn, for that introduction, and hello to all of you who are attending this webinar. So today we're going to be talking about how to choose your super resolution technique. And so here's a quick content overview for everything that we're going to be reviewing today. So we'll go over the current technology landscape, just kind of reviewing current techniques in the field. We'll give you an introduction to single molecule localization microscopy so that everyone can become familiar with the technique. We'll then review how to choose your best technique and also some sample preparation considerations, and also the question of how do I get started. And then of course at the end we'll have a Q&A for those of you who have questions about anything that we covered today. But before we begin, I just wanted to get an understanding for where everyone is in their super resolution journey. So if you could just give a quick answer to these questions just so that we can gauge your knowledge base, that would be great.
I'm just going to leave it up for another second or two, Laura and Vanessa, because it looks like we have 79% of people have voted, now 84%. All right, and I will now close the poll and just share the results.
Okay. Carolyn, are you able to see the results on your screen?
Yes. Are you able to see them? It says 44% are new to super resolution. 25% says I've done some training, but I'm not a confident user yet. And 31% say I'm confident with imaging and I'm exploring new uses.
Okay, perfect. So this is great. A lot of you seem to be new to super resolution, so we'll go over all of those techniques now. Okay, so here is just an overview of the current technology landscape. So here I just wanted to kind of go over all of these techniques and some of the strengths and limitations of each. So we have electron microscopy, which is fantastic if you want to get nanoscale resolution, down to maybe 10 nanometers below that. And some of the cons, though, to this technique is that you can't do this in living cells. And if you wanted to label your different structures with some fluorophores, it'd be difficult to do that just because of the way that this technique is contrasted. Over here in the middle, we have confocal, which I'm sure everyone is very familiar with. Confocal is great for imaging a broad range of sample types. You can look at cells, tissues, and you can just get some really great optical sectioning and 3D renderings with this technique, though you are limited in the resolution as well. So if you wanted to look at proteins or different receptors, it would be difficult with the confocal. And then over here we have light sheet. Light sheet is also great if you wanted to look at 3D structures like organoids and just get a representation of that sample. Though if again you wanted to look at 20 nanometer resolution and maybe see some proteins, it would be difficult because of the diffraction limit of light. So here on this next slide, I have wide field imaging, which I'm sure many of you are also very familiar with. Wide field imaging has a broad field of view and is compatible with a lot of different tissue samples, as well as some live cell imaging. And it has an adjustable illumination angle so that you can, get a better signal to noise ratio as you're imaging with this technique. But the three that I have listed here are epifluorescence, high-low, and turf. So with epifluorescence, you have light that's coming in at a 0-degree angle, and this illuminates your entire sample. So you get both in focus and out of focus light. This is great if you have some thicker tissues, though if you have tissues that are maybe just 10 microns beyond the surface, then that's where high-low would come in. This is just another adjustment to that illumination angle so that you can see with more clarity and just a better signal to noise ratio your sample. And then we have turf, and turf is basically changing your illumination angle of light to what's called a critical angle. And this allows you to see structures at the cover slip. And this is where single molecule imaging ties into play with turf so that we can image right at the cover slip and we're able to get those localizations. Turf is not a localization technique, but it does assist in getting a better signal to noise ratio, essentially. So some of you may already have a nanomovager, but for those of you who don't, I just wanted to give a brief overview about this technology. So the way that we beat the diffraction limit here at ONI is with the nanoimager, and we do this with super resolution, specifically single molecule localization microscopy. And this platform is very unique because it is compact. It's very different from microscopes that you're used to seeing, as this footprint is smaller than a sheet of paper. This is also very robust, so you don't need an optical table or a dark room to use this instrument. It has some vibration pads, so it can just sit on any bench top. It's also very easy to use as there is not really any hardware, so it's a quick start and you don't have to align. And it's also very flexible and incorporates all of those different illumination modes that I had mentioned in the previous slide. So it can do epifluorescence, high-low, and turf, and you can do fixed or live cell imaging on this platform. So this is one box with many techniques. So here's just an example of a few of them. This isn't everything, but it does kind of give you an idea. So with this nano imager, we can do destorm, palm, and paint. And these are fixed imaging techniques. Palm can be live, but essentially you can resolve and visualize structures down to 20 nanometers. We also have the capability to do single particle tracking. So if you wanted to do a live cell experiment to look at single particle dynamics and their behaviors, you could do that. And then you can also do single molecule FRET to look at different protein interactions at the nanoscale.
Yes, so before we dive into choosing your microscopy technique, I want to have a little recap on what single molecule localization microscopy is and where can it potentially fit in your research, okay? So if we have a look at this diagram, we see the biology at different length scales. So you could be interested in imaging small molecules all the way up to imaging big animal tissues, such as a mouse brain. And depending on the size of your structure of interest, you'll probably want to use different microscopy techniques. So for example, if we go to the bigger length scales, we would have light sheet microscopy, as I already mentioned, or confocal or SIM, which would enable you to visualize cells, to visualize organelles, organs, or even whole microorganisms. But to have a look at smaller structures, so below the diffraction limit, one would need to use super resolution microscopy techniques. In this category, we find techniques such as STET. We found the single molecule localization microscopy, or SMLM, and we find novel advanced microscopy techniques such as MinFlux. And if we go a step lower, we also have here for comparison electron microscopy, which is of course not based on light, but on electrons to perform your imaging. So today we want to focus on SMLM, on single molecule localization microscopy, and why is it beneficial and how can you apply it to your research? So in first instance, if you have a look at your structure of interest, here we display a cell that is labeled with fluorescence. In this specific case, some cell receptors, so surface markers, were labeled. With conventional fluorescence microscopy, such as epifluorescence, yes, we can detect our cell. We can conclude this is our cell, it's roundish, it has some intensity, so it's probably our signal. But we are very limited in really identifying nanoscale features, and we can also be limited in sensitivity when our labeling or our signal is very low. This is where single molecule localization microscopy comes in, because in essence, it gives you a 10 times better resolution compared to standard fluorescent microscopy. And this is just a number, 10 times, but how impactful it is you see with this image comparison, where with super resolution technique, you can start seeing really the outline, these nice protrusions of the cell, and you can start seeing the nanoscale spatial distribution of these receptors, with the receptors localizing, having a lot of signal at the outside and a little bit less in the middle of the cell. This will allow us, because it's a localization microscopy technique, to also quantify this biomarker of interest. And all in all, with super resolution, the final goal is to get a deeper understanding of your sample and your biology. So how do we achieve super resolution and specifically single molecule localization microscopy? This is my only technical slide, I promise, but we have to understand the diffraction limit of light before understanding how we can overcome it. So I have a very, very simplified equation for you here, very simplified, and it's basically the diffraction limit of light. The limit we can go with conventional microscopy is approximately half the wavelength of the light we're using. Okay, so this is normally between the ranges of 250, 300 nanometer. What does this mean? That structures that are smaller, for example, structures that are 100, 50, 10 nanometers apart, we will not be able to distinguish. We will see as one structure, as a big blob of light. What else is the limitation? Not only we cannot distinguish, but for example, a structure that is 10 nanometer in size or 100 nanometer in size, we would not be able, based on the signal, to distinguish which is which. So everything below that diffraction limit will appear as a big blob, and we will not be able to distinguish nanoscale features. How does single molecule localization microscopy come into this? we are not breaking any physical laws. Physical laws are what they are, but some scientists discovered a way to circumvent them. Okay, so instead of imaging your sample, looking at all the fluorescent intensity at the same time, actually the trick here is to divide the fluorescence in time. Okay, and this is done by selectively illuminating or switching on one fluorophore, localizing it, switching it off. The next one, switching it on, localizing it and switching it off, and so on and so forth until we localized all the fluorophores in our sample. Okay, and our final image will be a reconstruction of all the dots we localized. If we zoom in into how this localization process works, so once we detect the fluorescent signal, we localize it, and we do this by doing a mathematical fitting until we find the position of this fluorophore and can add it to our position list. If we sum this up over many frames, we will get a beautiful reconstruction of our sample, and this is what allows us to obtain this 20 nanometer resolution. How does it work in practice? Okay, this was a bit of theory. In practice, what we will want to see or what we want to obtain is an image like this. This is again an example of a cell where the fluorophores are activating, are switching between their on and off state. So you will see, as a result of this, you will see that your sample is blinking. This blinking is our goal and what we want to achieve to obtain this high resolution imaging. At the end, when all the localization are reconstructed, we get an example image like this one on the right side with a resolution of 20 nanometers or below. And you're not only doing science, but you're doing nice pointillistic art in the same goal. So this was an introduction to how to perform the D-storm microscopy, but what about the quantitative part? Okay, so from our localization data, we can extract certain features. We can extract biological relevant information. This is normally performed in the field with analysis such as clustering analysis. Here I have an example again of a cell where the surface was stained, and we are interested in clustering these surface markers. For that, at ONI, we offer the software CODI, which stands for Collaborative Discovery, which will help you to analyze your single molecule localization data in a very intuitive fashion. And once the clustering is performed, okay, we're not only getting nice images again, we're getting quantitative output. So we can get information such as cluster size or the number of localizations that we detected per single cluster. And so now I showed you a lot of cell examples, but of course this also can be applied to a broader range of applications such as nanoparticles, in which maybe you're interested in investigating nanoparticle cargo or nanoparticle outside functionalization, or even viruses or EVs. You can go a bit deeper into the cell in really investigating organelles, cell organelles, so to understand how is there any change in morphology, is there any difference between disease state and healthy state, and all the way up to tissues, again, to maybe have some biomarker discovery, or to understand the influence of a specific drug on your tissue at the level of tissue organization. And I have talked a lot now about D-storm, okay, because D-storm is a very commonly used single molecule localization microscopy technique. And as I mentioned, we have the nice 20 nanometer resolution. We can even do multicolor, although it can sometimes be challenging because it depends on the dye you choose. And we showed you that you can do the quantification. However, we also have some limitations, such as it's limited to fixed samples because we need really high laser power, and we are limited by the number of fluorophores that have this photo switching behavior. Luckily, in our toolbox of SMLM, we have more techniques that we can choose from that circumvent a bit these limitations. One of them, which is really recommended for live cell imaging, is palm imaging. Okay, in this case, we have fluorophores that are in the off state, so they are not fluorescent. Your sample is dark, and they become fluorescent stochastically, so randomly, once you activate them with a light input, normally with UV light. You activate your sample, you can detect your fluorophore or even track it in time, such as this example with live bacteria. The second option in our toolbox is paint modalities, and the most famous one probably being DNA paint, in which this is completely independent on the dye photophysics, but how to achieve this blinking behavior is by reversible interactions. In the case of DNA paint, this blinking behavior is achieved with DNA-DNA interaction. Okay, when you have your one single strand of DNA on your structure of interest, for example, your secondary antibody, and then a second one complementary to their structure with the fluorophore, which binds and unbinds again to your target strand. Once it binds, we are able to detect them. When it unbinds, because of DNA hybridization, we are not able to detect them anymore. And here we have this example of clatrine pits. So I have hoped to give you a good overview of SMLM, and now I'm giving the floor back to Vanessa to discuss with you how to choose your best technique.
Yep, thank you, Laura. So now that you know a few of the single molecule localization methods, we'll then go into how you can choose the best technique. So we have a roadmap to just help guide you through choosing the best technique. And one of the questions that you want to ask yourself is, what is the size of my structure? If it sits right below the diffraction limit of light, you have a couple of super resolution techniques to choose from. And the next question you want to ask is my sample live or is it fixed? If it's live and you're right below the diffraction limit of light, you can use a method like SIM to help you visualize cytoskeleton dynamics. And if your sample is fixed, then you can use a super resolution method like STED, which will help you visualize cell receptor clusters. But if your structure is smaller than 100 nanometers, then you will want to utilize a single molecule localization technique. So for live cell imaging, for example, to monitor the transport of a protein walking along the ER, or for watching a subpopulation of UVs being uptaken into a cell, Palm will be the best technique for you to visualize those events. And Palm does require photoactivatable and photoswitchable dyes that can localize single molecules, and you don't need a buffer to do this method. So if your sample is fixed though, then D-storm will be optimal for viewing structures like extracellular vesicles, LNPs, nuclear pores, and viruses. And if you want to multiplex your sample, then that's where DNA paint will come in, because you can do more than 3 colors. And it basically, as Laura mentioned, it utilizes the transient binding events, mainly between DNA strands, to localize single molecules with high precision. So we're just going to go into a few examples for how to choose your best technique. So in this example, we're looking at EVs, or extracellular vesicles, and EVs are released by all cell types, and they actually encapsulate various molecules, including proteins, DNA, RNA, metabolites, and lipids. And these vesicles just circulate throughout our biofluids, and they can be uptaken by neighboring cells. And the contents of these particles are involved in a lot of intracellular communication and can affect the function of the recipient cells. So because of this, EVs have a great potential in diagnostics and in therapeutics. But for this, we're just going to follow the same roadmap that I had on the previous slide. So you ask the question, what is the structure of my sample? In this case, EVs are usually around 30 to 100 nanometers. Some of them can be larger, but on average, we'll say 100 nanometers. So we know that. And then we ask the question, is this going to be live or is this going to be fixed? And in this case, maybe we want to look at a fixed sample. And so in this case, we'll be using D-storm. So here's just a slide to kind of go into analysis for what you can do when you acquire a D-storm experiment. So for this, data was acquired with CODI, which is our collaborative discovery platform that Laura had gone over a little bit previously. And Masako Harada's lab was looking at EVs that were isolated by tangential flow. But the main goal of this experiment was to understand two different populations of EVs in their samples, so small EVs versus large EVs, and pull out biomarker distributions to see if there were any changes among these two sizes. So the clustering parameters were the same for both these small and large EVs except for the area, and this was just to separate out those populations. So they discovered that this biomarker X actually had a higher expression in smaller EVs versus larger EVs, and this is just an example of how you can look at a novel marker and how it might be expressed differently, due to biogenesis or just due to different targeting. All right, in this example, we have a palm experiment. So in this application, we're looking at cohesion proteins labeled with photoactivatable Genelia fluor. The fluorophore is 549. So if you want to perform a palm experiment, you need to first identify the protein that you're interested in, and then you'll want to fuse that protein with the photoactivatable fluorophore. In this case, the size of this cohesion protein fits that 10 to 100 nanometer size range, and this is live. So now we can do experiments like single particle tracking, and with this we can figure out if a molecule is immobile or how many of them are actually diffusing. So for this, if you wanted to say multiplex your sample, so if you had an experiment where you wanted to do more than 2 or 3 colors, then you would want to choose DNA paint because this is best for multicolors and you're not limited to your dye options. Though if you wanted to do an experiment with just 2 colors or even 3 colors, then D-Storm and Palm will be perfect for that. Typically on an instrument, you'll have 2 to 3 lasers that you can use to do an experiment such as this. All right, so now that we reviewed how to choose your best technique, the next consideration is sample preparation. And sample preparation is very important when you're doing any sort of single molecule localization experiment, just because there are a few things you need to consider. So a lot of people, ask these questions of how do I fix my sample? Which antibody concentration is good to use for D-Storm? Is my commonly used fluorophore even compatible? And how do I know if my final image is good quality? So let's go over all of these. So first, let's look into how do I fix my sample? So to choose a fixation method, if you require, you need to first determine if you require a mild fixative or if you require a strong fixative. So for mild fixation, this is optimal for those who are studying intracellular non-membrane proteins, such as nuclear proteins and cellular organelles. Exceptions are structural proteins such as tubulin and actin, and these are optimally fixed with a stronger fixative. There are cases where PFA, which is a mild fixation, can be insufficient to cross-link many of these membrane proteins, and this can just lead to antibody-induced protein clustering. However, a stronger fixation better preserves the native structure of your sample, and it enables you to confidently measure this sort of membrane protein clustering. In our case, with the discovery kit, we do have glutaraldehyde included in the kit, which is a strong fixative to help you if you needed that sort of reagent. All right, so let's just go into an example. So here's a case study where the ONI discovery kit was used. So on the left-hand side, you can see that a nuclear protein histone and a TOM20 protein that plays a crucial role in the translocation of proteins into the mitochondria were stained with the standard fixation. So here we have CF583R and we have TOM20 labeled in AZ647. So in this condition, a permeabilization step was used to stain these intracellular targets. Though if we were just looking at an extracellular target, then we wouldn't require a permeabilization step. On the right-hand side, we have a lysosomal protein that's labeled with CF583R, and then we have CD63 that's labeled with AZ647. These are membrane proteins, so we did use PFA plus glutaraldehyde to preserve the native structure of these proteins. But it is important to note that not all proteins can be fixed with glutaraldehyde, as it can disrupt some of the epitopes. So we do recommend that you search the literature and just ensure that your fixation conditions have been optimized for your protein of interest. All right, so the next question that people always ask is, oh no, sorry, this is the antibody concentration. So which antibody concentration is good to use for DStorm? So it's important for the full structure to be fully labeled. And so what I mean by that is if a continuous structure is not labeled with enough efficiency, it will appear as punctate in your DStorm image. So you can see that here. This is an example of a low label density, and because of that, this is what your image will look like. There are a few gaps between the localizations, and you don't have a clear picture of your original structure. So this is because in diffraction limited microscopy techniques such as confocal wide field, labeling density isn't as critical because the diffraction limit of light actually masks these gaps, so it's easier for you to see your structure. Though because we're going down to such a low resolution, a lot of things are going to be unveiled. I mean, you can even see free antibody in your sample, so it's very important that you kind of optimize these techniques so that you have a continuous structure. And to that, if you also want to ensure that you don't have this concentration in excess, as this can induce a high background signal. So which antibody concentration is good to use for DStorm? We recommend increasing the primary antibody concentration that you'd normally use for confocal and wide field to 2 to 10-fold. And we also recommend a direct labeling method such as primary antibodies, halo tags, to ensure that the fluorophore is close to your target. This is because when you label structures with a lot of epitopes, there can be some steric hindrance. So you just want to make sure that your fluorophore is very close to the structure that you're interested in imaging. Okay, so this is the question: is my commonly used fluorophore compatible with DStorm? The quick answer is it depends. So if you have specific fluors like Alexa Fluor 647, or if you have Atto 488, then yes, those will be compatible. But the reason why is that the photophysical properties of fluorophores can influence the quality of your super resolution image. So two important considerations are the number of protons and the duty cycle for blinking. So a high number of protons emitted per blinking event yields a greater localization precision. So that means you'll always want to choose a fluorophore that's very bright. And the duty cycle is basically the fraction of times that the fluorophore is in the on state. So a low duty cycle makes it easier to distinguish the maximum number of fluorophores within a diffraction limited area, but you want to have a fluorophore that blinks on and off so that you can localize its position spatially. And you can see that example here. So here we have high photons and a low duty sample. So we have these fluorophores that are blinking on and off effectively. And so we're able to reconstruct this image and see, what it looked like initially. But over here in this example, we have an object, and maybe we used a fluorophore like GFP. And so now we have low photons and a high duty sample. So that means that maybe this fluorophore didn't blink enough times, and so we couldn't capture all of those localization events. And so that's why it's really important to choose a fluorophore that's compatible. We do have a list of fluorophores for DStorm and Palm. That's on our website. Also, if you just want to reach out to one of your representatives, we can also send you that list as well. And then the final question, how do I know that my final image is good? There are a few things that you need to keep in mind. First and foremost, you want to have imaging controls. And it's really important that you have a negative control or even an isotype for your antibody, just so you know what's real signal and what's not real signal. Another way that you know what's good is just through experience. So we do have our DStorm training kit. This is actually really nice because it lowers the barrier to entry. And with this kit, we also have a video, so you can basically follow along straight from the sample preparation to image acquisition to even analysis. And you can really use this training kit on any system. It doesn't have to be on the Nano Imager, just as long as you have access to a super resolution microscope. And then the last question is my structure reconstructed fully? So this goes back to what I was mentioning earlier, where you just want to make sure that you have fluorophores that are compatible with DStorm, so they have a low duty cycle and a high photon count. And then you also want to make sure that you have a high label density so that you don't have any gaps in your structure whenever you're imaging. All right, so now I'm going to pass this back to Laura, and she's going to talk to you about how you can get started with the super resolution experiment.
Yes, fantastic. I hope we have inspired you to choose the best technique that's suitable for your research. And how do you get started now? Okay, so this is the most typical question, and you can watch many webinars, and we encourage you to learn more, but we also believe that the best way to get started is to get hands-on experience on the microscope, on the sample prep, and on the imaging. For this purpose, as Vanessa already mentioned, ONI is offering and developed this D-Storm training kit, because this is an easy way to get a super resolution sample and to get hands-on experience in how a storm image looks like, and more importantly, how do I analyze the storm image, which is a very big burden sometimes for some of the researchers. So this is just what the kit looks like. If you order this kit, it will come with everything, so even with your fixed cells, and it will give you some reagents to stain your structure and the imaging buffer that are required for storm. If you prefer, you don't need to do this on your own, but you can also join the live from the lab sessions under the guidance of one of ONI's application scientists. Now, there are some of you who have done some microscopy but are not feeling really confident. We have a second level of reagents, which is when you're ready to label and image your own structure. We developed the ONI Discovery Kit for you. And the nice thing about this kit is that it comes with two color D-Storm secondary probes, okay? So if you are a bit familiar with D-Storm, you will realize that obtaining two fluorophores that are blinking very nicely in the same buffer is not an easy task. So here, our research and development team has done a great job to find you the best storm fluorophores that are blinking nicely with the ONI buffer. And of course, if you need some further guidance, such as fixation, we also advise you on that and provide you with the fixative and the labeling reagents and permeabilization if you would need it, and again, the imaging buffer. Finally, I just want to mention that we also have an application-based kit, which is the EV Profiler kit, as Vanessa mentioned before, where you could perform some nice imaging and analysis on your extracellular vesicle sample. Okay, and in this case, this kit is specially designed to capture your vesicles on a ship, to label them, and again, to image them with our D-Storm buffer. And what is the nice thing here, that this kit also provides 3 biomarkers already, but if you have your own biomarker of interest, you can easily swap it if you have a compatible fluorophore. And it allows you, because of the protocol guidance, to image thousands and thousands of EVs per field of view and get information about the biomarker distribution at a single vesicle level. And I want to spoiler alert a little bit. Our research and development team has done a lot of effort to bring you a new version, EV Profiler 2, in the next coming month. So stay tuned. So with this, I want to thank you so much for your attention. And I want to again highlight that we have a lot of resources on the website, oni.bio. If you go into the application section, we can have resource sources such as this webinar, which will be uploaded later on. We have application notes, some advice on fluorophores. If you're unsure, is my fluorophore compatible? You can find the list of compatible fluorophores, etc. From the ONI part, we hope we are working for you to make imaging, especially super resolution imaging, easier, to make it more accessible, and so you can image, you can visualize and analyze your data faster, so you don't need to learn a lot of the details, but can jump straight into the results. And that is our mission, and we will continue working for it in the future, so stay tuned for later developments. With that, I think we're ready to open the Q&A sessions. So I'm very curious if you have any question about your sample or about choosing the best technique for you.
Wonderful. Great. Well, thank you very much for the presentation, Laura and Vanessa. Okay, we're now going to open the floor to some questions for Laura and Vanessa, which they will be answering live. And once again, if you have any questions, please feel free to drop them into the chat box. And it looks like we already have some submitted. So for the first question, we have, can you use all techniques on cells other than animal cells, i.e. plant, bacteria, yeast? Are there any techniques that work better in different cell types?
Laura, did you want to take this one? Did you want me to?
I can take this one. Yes, so your cell types, what is important, especially if you want to do localization microscopy, is that it's attached as close to the surface as possible. Because as Vanessa already mentioned, normally with these techniques such as STORM and DNA paint and PALM, we're using turf illumination. So we to get a better signal to noise ratio. So what is important here is how you want to attach your structure to your microscopy cover slip. And there are different strategies, and it's not really depending on the origin, but a bit depending what is your structure. So if you have an EV, we have a special protocol to attach them to the surface with capture molecules, but the same if you would have an EV from a plant or a bacteria source, you would have a special surface treatment to capture your structure of interest. The same for bacteria and virus plants. So normally research with bacteria, I know some people are really interested in imaging live bacteria. So here the question would be to use PALM for live, but you can as well use STORM if you have fixed cells of a fixed sample. In general, there is no limitation in terms of source, but it needs a bit of optimizing on the sample preparation side. But I think more important is the question if your tissue or your cells is live or if it's fixed. Then yes, you would have to choose a little bit what is the best technique.
Great, wonderful. Thanks so much for that answer, Laura. All right, so the next question is, how beneficial is it to use a labeled primary antibody rather than primary and secondary? Or is it even better to use nanobodies?
Right. It's important. It does help when you have like a primary conjugated antibody, just because the distance is closer to your structure of interest. When you do a primary secondary labeling, it's a bit further. And so especially when we're going down to the single molecule level, you do want to have accurate precision and a good representation of where your protein or whatever it is that you're labeling is located. But if you are able to have a nanobody, great, use that too, but we definitely recommend primary conjugated over primary secondary.
All right, wonderful. Okay, next question is, what do I do if I have more than 3 targets in the samples?
Yes, that's the tricky one, right? So for more than 3 targets in the samples, as Vanessa already hinted, it's rather challenging because normally you are limited by the number of laser lines and filters in your microscope. Okay, and also especially for this STORM, it's not so easy to find more than 3 compatible dyes that have this photophysics that link. So for this purpose, there is a whole research going on into paint approaches, especially DNA paint, because DNA is very flexible in the design. With the base pairing, you can play a lot, and depending on, for example, parameters such as kinetics, binding kinetics, you can have different. You will not see different colors, but different pairings, different DNA pairings. And this flexibility and still this specificity of DNA allows you to have more than 3 colors if you wish. I think in localization microscopy, It's the standard for more than 3 to use a DNA DNA paint kind of approach. I also have to say it is quite a challenging endeavor, so don't be frustrated. DNA paint with more than 3 colors is definitely a very advanced research question. So you'd always recommend, yes, to start with few and then build up.
All right, I've got a few more questions in. So for the next question, what to do if no good antibodies are available?
If no good secondary antibodies are available. So we do, okay, so for DStorm, Alexaflor 647 is a pretty popular secondary antibody. That one I feel is pretty accessible, though if you still have issues finding it, our discovery kit does have a couple fluorophores that are compatible with DStorm, so CF583R and that 647. But usually you could also get away with some fluorophores like Site3, that's also known to blink. So you're not extremely restricted to what fluors you can use. It's just that, you know, your bucket for what you can choose from is a little bit more limited with a super resolution technique like DStorm or Palm.
Okay, next question. Can permeabilization interfere with my imaging?
That's a good one. So for permeabilization, of course, when you have intracellular structures that you want to visualize, it's a must, no? Because normally our antibody does not go inside of the cell without the permeabilization step. Now, it doesn't necessarily interfere with the imaging because there are some washing steps afterwards, but what it can interfere with is with background, okay? So normally with permeabilization, we always recommend as a general rule to have the condition with permeabilization and without permeabilization, so you make sure that what you finally image is actually not just a random background, but it's really specific, your sample of interest. So in that way, having good control can prevent that and can give you more confidence that the permeabilization worked and that the permeabilization is not hindering your imaging. But in general, it's okay to use, especially for interior cell targets.
Okay, great. So on to our next question, and this one is from Arshad, and he asked, which super resolution microscopy techniques are ideal to observe DNA damage and to track molecules in these cells?
Yeah, I don't have a lot of experience with DNA damage, but I heard tracking. So I'm gonna jump in. So normally what you want to do in this case is single molecule tracking, okay? And you can do that also with the nano imager or with turf illumination. And the thing here, it's not strictly speaking, it doesn't give you the 20 nanometer resolution, okay, but instead of the resolution, what you gain is the dynamic information. So if you have a sparse sample, it's important to have a sparse sample so you can distinguish individual molecules. You're able to detect this fluorescent molecule and track it in time. As an output, what you will have is a diffusion map of your molecule, and probably depending on what you label, if you label the membrane, you will have, because you have all these tracks, you will have an outline of your cell. Or you can have, if you observe biological phenomena, okay, you can have information about the speed. So if it's taken up by a cell, does it slow down? Or if it interacts with this organelle, does it slow down or speed up? So that's a nice complementary technique which one can use, and this is very compatible, which what we mentioned with palm dyes, palm type of experiment.
Wonderful. All right, so we have Suzanne who asked, is it possible to track viruses of 25 nanometers in size with 5 FPs on its surface?
5 FPs on the surface. You can track viruses, yes. You can do palm single particle tracking. So you can do single particle tracking 2 ways. You can do it in wide field. You can put your illumination mode into turf and track them on the surface, or you can do it in super resolution, which is palm. You just have to adjust your sample preparations to include a palm fluorophore. But for your virus that's at 25 nanometers, yes, you can do that. For the 5 FPs, is that like the amount of like epitopes and how much you want to label the virus? In that case, I would think DNA paint would be the best method to do that so that you can have multicolor labeling and multiplex.
Yeah, then it sounds like it's a low labeling 5. So you want to make sure that if you label your virus, you have a really, and you track it, of course, you have a really, really bright dye, okay? Because yes, we can detect single molecules, we can detect one single fluorophore, but it has to be rather bright in intensity. And again, there are some recommendations in literature on how to choose your brightest molecule for tracking purposes. Yeah.
Okay, great. Right. So the next question is from Sadiq, and they are asking, do you have any experience with samples which are prepared using click chemistry tool? For example, I am, oh, sorry, someone is in the document and covering. For example, I am clicking my probe with Sci5 in fixed cells. So is it possible to use SMLM?
Yes, fantastic, fantastic experiment. I believe one of the early D-storm research out there was actually done with click chemistry on cell membranes. And the important thing here is again the choice of dye, with Sci5 being compatible with this storm. So in principle, this would work with the correct storm buffer. So once you add your buffer to obtain this photo switching behavior, this would be very compatible with this storm. I would say yes.
Wonderful. All right, so the next question is from Sandra. Sandra is saying, what about calcium imaging? So can you offer any input on calcium imaging?
So for calcium imaging, you could do calcium imaging on the nano imager for sure. We have a camera that can capture those events pretty quickly, just because it's not a PMT, it's an actual detector, like a camera detector. So we can get down to those frames per second. As far as doing this with Palm, have you done this, Laura, before, where you have you seen it before with calcium imaging?
It sounds familiar, but I don't know the experimental details now, but I think I've seen it. So what I would recommend is if you go to our website on i.bio, you will also see a section where we have publications that were done with the nanoimager. So I'm pretty sure there was one with calcium imaging, but I would have to check and maybe come back to you.
All right, wonderful. So then we can come back to Sandra. And then the next question is from Karsten, and Karsten is asking, what are available laser wavelength and what are typical laser powers?
Right, so the available ones on the nano imager are 405, 488, 560, and 640. And as far as laser powers, so for D-storm, you want high laser powers. Ours go up to around 250 to 300 milliwatts at 100%, though you can, use probably like 50% of that when you're doing your D-storm experiment. But you just want to make sure that, yes, you have enough output at the fiber for you to do this experiment.
Right, excellent. So it looks as if we are at the end of our questions. And we are almost at time. So a very big thank you for the presentation again, Laura and Vanessa. And if you were unable to get to your, if we were coming back to some of the questions, I will make sure that Laura and Vanessa are able to come back to you specifically. But once again, this webinar was recorded. Therefore, if you need to refer back to it, you can. And all of these recordings can be found within the resources section of our website. So make sure to check that out. And thank you so much to all the attendees for joining us today. And upon exiting the webinar, you will receive a survey. So please fill out that survey. It takes 2 seconds and it's going to provide us some feedback on your experience today, or tell us if you're in need of being contacted or how we can improve these webinars for everyone on this call. And thank you again. And we hope that everyone has a really wonderful day. And thank you again to Vanessa and Laura. Bye.
Bye, thank you.